the molecular classification of gastric cancer, exploring the genomic landscape and its impact on treatment. Welcome to today's podcast of the Everything Surgery podcast, where we delve into the fascinating world of gastric cancer and discuss the transformative role of molecular classification. Traditionally, gastric cancer has been classified based on its histological appearance, such as the intestinal and diffuse types, and by its anatomical location, whether in the cardia or non-cardia regions. However, recent breakthroughs have shifted our approach, focusing on the genomic drivers that define distinct subtypes. This evolution is crucial for both prognosis and the selection of targeted therapies. Let us begin with one of the most influential frameworks, the classification proposed by the Cancer Genome Atlas Research Network in 2014. This comprehensive study analyzed nearly 300 gastric adenocarcinomas and identified 4 major molecular subtypes. The first group includes tumors that are positive for the Epstein-Barr virus. Approximately 9% of gastric cancers fall into this category. These tumors are characterized by the presence of viral DNA and RNA within their cells, which leads to extreme DNA hypermethylation. This process silences numerous tumor suppressor genes, particularly those affected by the so-called CpG island methylator phenotype. A notable feature of these tumors is the presence of recurrent mutations in the gene known as phosphatidylinosytol 4,5-bisphosphate 3 kinase catalytic subunit alpha, a key player in growth pathways. Additionally, there is amplification of genes such as Janus kinase 2 and programmed death ligand 1 and 2, which can promote immune activation or evasion. These tumors are more common in males and typically found in the fundus or body of the stomach. They often display intense lymphocytic infiltration and generally have a more favorable prognosis than other subtypes. Importantly, the high expression of programmed death ligand 1 makes these tumors suitable candidates for immune checkpoint inhibitor therapies, such as treatments targeting programmed cell death protein 1. Moving to the second group, we encounter tumors with high levels of microsatellite instability. Roughly 22% of gastric cancers exhibit this feature. These tumors have a deficiency in the DNA mismatch repair system, resulting in a very high mutation rate and frequent frameshift mutations in genes with repetitive DNA sequences. Tumors with high microsatellite instability tend to have a favorable immune contexture, owing to their abundance of tumor-infiltrating lymphocytes and neoantigens. These cancers are more common in the distal stomach and are typically associated with the intestinal histological type. Patients with this subtype often have a better prognosis and are especially responsive to immunotherapies. For example, pembrolizumab is approved for advanced cases of gastric cancer with high microsatellite instability. It is worth noting that these tumors may respond less to certain chemotherapies, such as fluorouracil. The third category comprises tumors that are genomically stable, accounting for about 20% of cases. These tumors have a relatively low number of somatic copy number alterations and mutations. A recurring feature is the presence of mutations in the ROS homolog family member A gene, which plays a crucial role in cell motility and adhesion. There are also unique fusions involving claudin 18 and Rho-GT-POS-A activating protein, which disrupt cell adhesion and signaling. Genomically stable tumors often harbor mutations in the cadherin-1 gene, responsible for hereditary diffuse gastric cancer and the loss of E-cadherin function. This subtype is strongly enriched for diffuse-type histology, including signet ring cell carcinoma, and tends to occur in younger individuals. Unfortunately, it is associated with poor prognosis and often presents in advanced stages with peritoneal spread. Recent advances offer hope, such as the development of monoclonal antibody therapies targeting claudin 18.2. The fourth and most prevalent group is marked by chromosomal instability, representing half of all gastric cancer cases. These tumors display a high degree of aneuploidy and frequent amplifications of receptor tyrosine kinases. There are recurrent mutations in the tumor protein page 53 gene and focal amplifications of several oncogenic drivers, including human epidermal growth factor receptor 2, vascular endothelial growth factor A, epidermal growth factor receptor, mesenchymal epithelial transition factor, fibroblast growth factor receptor 2, Kirsten-Rat sarcoma viral oncogene homolog, and cyclin E1. This subtype is strongly associated with the intestinal histological type and tumors of the gastroesophageal junction, being most common in Western populations. Treatment options include therapies targeting human epidermal growth factor receptor 2 and vascular endothelial growth factor. such as trastuzumab and ramucirumab, respectively. Ongoing clinical trials are examining other targeted agents as well. Let us now turn our attention to an important refinement from the Asian Cancer Research Group. This classification further divided gastric cancer into 4 groups. Tumors with high microsatellite instability, which have the best prognosis. Microsatellite stable tumors with an epithelial mesenchymal transition signature, which carry the worst prognosis and are similar to genomically stable tumors. And 2 intermediate groups based on the presence or absence of tumor protein page 53 mutations. To summarize, molecular classification has become essential in prognostication and guiding therapy beyond traditional chemotherapy. This approach identifies patients with both favorable and poor outcomes and allows for the selection of targeted therapies. For example, immune checkpoint inhibitors for Epstein-Barr virus-associated and high microsatellite instability tumors, anti-human epidermal growth factor receptor 2 therapies for chromosomal instability tumors, and anti-claudin 18.2 monoclonal antibodies for genomically stable cancers. Routine testing for mismatch repair, microsatellite instability, human epidermal growth factor receptor 2, and programmed death ligand 1 is now standard practice for advanced gastric cancer. Additionally, testing for claudin 18.2 is rapidly entering clinical practice. Thank you for joining us on this journey through the molecular landscape of gastric cancer. Your feedback and questions are always welcome. Until next time, it's the Everything Surgery Podcast. Dissect the extraordinary.